Welcome back to the deep dive where we, break down these complex topics just for you.
Yeah.
And today we're going on a journey, I'd say, into the fascinating world of Class A rhodopsin-like receptors.
Okay.
And those are the largest subfamily within G protein-coupled receptors.
That's right. And they are essential to so many processes. We're talking vision, smell, radiolating, heart function, immune responses.
Right.
And they're a major target for a huge range of diseases.
Wow. That's a lot to cover today.
It is.
So we're going to, I guess, dive deep into their structure, the function and relationship, and how drugs can target them.
Absolutely.
But get ready, because we're about to go into some intricate molecular machinery.
Yeah. And the best way to understand Class A GPCRs is to start with their structure.
Okay.
So imagine a protein woven through the cell membrane 7 times, forming a bundle of helices.
I'm imagining it.
The 7 transmembrane helix structure is their hallmark.
7 helices, got it. Yeah. And I remember reading about the first crystal structure of a GPCR ever solved, which was rhodopsin back in 2000.
Yeah.
Why was that discovery so significant?
That discovery was a game changer because it finally gave us a 3D view of how these receptors actually work. It was like having a blueprint, allowing us to visualize the intricate details of the transmembrane helices and understand how they move and interact with each other.
So before 2000, it was all theoretical.
Pretty much. We knew they existed and had an idea of their function.
Right.
But we didn't have that clear visual confirmation.
Oh, wow.
That rhodopsin structure opened up a whole new world for GPCR research.
Yeah.
And it's why rhodopsin is considered the prototypical Class A GPCR.
Okay, so rhodopsin is like our poster child for Class A GPCRs.
Yes.
What other celebrity receptors belong to this family?
Well, you've got the beta-2 adrenergic receptor, a key player in heart function.
Okay.
It's a target for drugs used to treat asthma and heart failure. So you could say it's a pretty big deal.
High stakes indeed. And you mentioned earlier that these receptors have 7 transmembrane helices.
Yes.
Are there any other structural features that are particularly important for their function?
Absolutely. There's helix 8.
Okay.
Which is interesting because it's not actually part of the transmembrane bundle.
Oh.
It often runs parallel to the membrane on the intracellular side.
So it's like a sidecar attached to the main structure.
A good analogy, but perhaps even more critical, are the loops connecting these helices, particularly extracellular loop 2 or ECL2.
ECL2. I remember that from the source material.
Okay.
What's so special about this particular loop?
ECL2 is essentially the receptor's gatekeeper for ligands.
Nice.
It's incredibly variable in length and amino acid sequence among different Class A GPCRs. And this variability is what allows each receptor to bind to specific ligands.
So it's like a customized lock for a specific key.
Exactly. The structural diversity in ECL2 has huge implications for drug development. Understanding the specific interactions between ECL2 and its ligands is crucial for designing drugs that can effectively target these receptors.
That makes sense. But to design those drugs, you need to know where the drugs are actually binding, right?
That's right. And the primary location for ligand binding pockets in Class A GPCRs is within the transmembrane domain, formed by those 7 helices we talked about earlier. Think of it like a pocket nestled within those helical bundles.
And I'm guessing the precise amino acid residues within these pockets are pretty important.
Crucial. In fact, they determine the shape of the binding pocket and how strongly a drug will bind to the receptor.
Right.
It's a delicate balance, like fitting a key into a lock.
I'm starting to get a real appreciation for just how intricate these receptors are.
They are.
But how does a ligand binding actually activate the receptor? What happens next?
Well, let's go back to our poster child, rhodopsin.
Yeah.
When light hits rhodopsin, it converts retinol, a molecule bound within the receptor, into an agonist. This triggers a cascade of movements, particularly in helices 5 to 7, which ultimately opens up the G protein binding site on the intracellular side of the receptor.
So it's like a domino effect with one movement triggering the next?
Exactly. And that opening of the G protein binding site is how the signal from outside the cell gets relayed to the inside, initiating a cellular response.
It's amazing how such tiny changes at the molecular level can have such a huge impact on the cell's behavior. But you mentioned ECL2 has a conserved disulfide bond.
Yes.
What role does that play?
Good catch. That disulfide bond is essential for maintaining ECL2's structure.
Okay.
Think of it like a tiny bridge holding the loop in the right conformation.
Okay.
Without it, the loop wouldn't be able to interact with ligands properly.
Fascinating. It seems like every tiny detail plays a crucial role in the receptor's function.
Yeah.
But I'm curious if ECL2 is so important for ligand binding. Does that mean drugs can also target this loop directly?
That's a great question and an active area of research.
Okay.
While most drugs target the transmembrane binding pockets, there's growing interest in developing drugs that can modulate receptor activity by interacting with ECL2.
Interesting.
It's a challenging approach, but it offers the potential for greater selectivity and fewer side effects.
This is getting really interesting. Can you give an example of how a drug interacts with a Class A GPCR?
Sure, let's stick with the beta-2 adrenergic receptor.
Okay.
Some drugs, called antagonists, work by binding to the receptor and blocking the binding site, preventing natural agonists like adrenaline from activating the receptor.
So they're essentially putting a roadblock in the way.
Exactly. Other drugs, called agonists, mimic natural agonists, binding to the receptor and activating it, triggering the same cascade of events as the natural ligand.
So it's like having a master key that can unlock multiple receptors?
Not quite. Remember that each receptor has its own unique binding pocket.
Oh, right.
So an agonist for one receptor won't necessarily activate another.
Right, of course. So it's more like finding a key that fits perfectly into one specific lock without accidentally opening any others.
Precisely, and that's one of the biggest challenges in drug development, designing drugs that are highly selective for their target receptor.
This has been an incredible journey so far.
Yeah.
I'm really starting to grasp the incredible complexity and importance of these receptors.
I'm glad.
But before we do, let's take a short pause to let all this new information sink in.
Sounds good.
When we come back, we'll continue exploring the world of Class A GPCRs. Stay tuned.
Welcome back. Ready to explore more about Class A GPCRs?
Absolutely. I'm eager to learn more about how these receptors are targeted in drug development.
Okay.
It seems like a daunting task, considering their complexity and the need for such precision.
It definitely is a complex process, but let's break it down step by step.
Okay.
Imagine you're a scientist working on developing a new drug for, say, migraines. You've identified a specific Class A GPCR involved in the pathway that triggers those debilitating headaches.
Okay.
What would be your next move?
Well, if I were that scientist, I guess the first step would be to find a molecule, a lead compound.
Yes.
That can interact with that receptor in some way, right?
Precisely. And that's where the real hunt begins.
Okay.
Scientists employ a variety of techniques to find these lead compounds. One common approach is high-throughput screening, where they test thousands, even millions of compounds against the target receptor.
Wow.
It's like casting a wide net to see what sticks.
Sounds like searching for a needle in a haystack, but with millions of haystacks.
Yeah.
How do they even begin to narrow it down?
They use sophisticated assays that can detect even subtle interactions between the compounds and the receptor. For instance, they might look for changes in the receptor's conformation. Or its ability to activate downstream signaling pathways. It's a bit like setting up a series of tests to see which compounds can talk to the receptor in the right way.
So let's say they found a few promising leads. What happens next?
Okay.
Do they just start testing those compounds in people?
Not so fast. The next phase is lead optimization.
Lead optimization.
Here, scientists use a combination of chemistry and structural biology to refine those lead compounds. Remember how we talked about those specific amino acid residues in the binding pocket?
Yes.
This is where that knowledge becomes crucial.
So they're tweaking the chemical structure of the lead compound to improve its fit into the receptor's binding pocket.
They might make subtle changes to the compound's shape size or charge distribution to increase its binding affinity, making it stick more tightly to the receptor. It's like tailoring a suit. You want a perfect fit for optimal effect.
I can see how that would be important for making the drug more effective.
Yes.
But what about safety?
Yeah.
How do they ensure that the drug doesn't interact with other receptors and cause unwanted side effects?
That's another key aspect of lead optimization. Scientists use techniques like structure-based drug design to visualize the interaction between the drug and the receptor at the molecular level. They can then make adjustments to the compound's structure to minimize the risk of off-target effects. It's like fine-tuning a musical instrument. You want to eliminate any unwanted noise or dissonance.
It sounds like a delicate balancing act, increasing efficacy while minimizing side effects.
It definitely is, and this process of lead optimization can take years, even decades,
Wow.
of meticulous research and refinement.
Okay.
But even with a highly optimized lead compound, the journey doesn't end there.
So what's the next step? Clinical trials.
You got it before a new drug can hit the market.
Uh-huh.
It has to go through rigorous clinical trials to evaluate its safety and efficacy in humans.
Okay.
This involves testing the drug in progressively larger groups of people,
Yeah.
starting with small, tightly controlled studies and gradually expanding to larger, more diverse populations. It's a multi-stage process, carefully designed to gather data on the drug's effectiveness, ideal dosage, and potential side effects.
It sounds like an incredibly long and complex journey from that initial discovery of a lead compound to having an approved drug available to patients.
It certainly is, but it's a journey worth taking.
Yeah.
Because developing new drugs that can effectively target Class A GPCRs has the potential to transform the treatment of a wide range of diseases.
You mentioned earlier that Class A GPCRs are involved in a vast array of physiological functions.
Mm-hmm.
Can you give some examples of how targeting these receptors can lead to new therapies?
Absolutely. Let's take the opioid receptors as an example.
Okay.
These receptors are involved in pain perception.
Right.
And drugs that target them, like morphine, are powerful painkillers.
Yeah.
But they also have serious side effects, including addiction and respiratory depression.
That's a classic example of the trade-offs involved in drug development.
Mm-hmm.
Powerful effects often come with significant risks.
Precisely. But by understanding the structure and function of these receptors in more detail, scientists are hoping to develop new opioid drugs that retain the pain-relieving properties while minimizing the risk of side effects. It's a major challenge, but one with the potential to alleviate suffering for millions of people.
I imagine that applies to other areas of medicine as well.
Absolutely. For example, researchers are working on developing new drugs that target dopamine receptors in the brain to treat Parkinson's disease.
Oh, wow.
They're also exploring ways to target chemokine receptors to modulate immune responses in conditions like autoimmune disorders and cancer.
It seems like the potential applications are almost limitless.
It does.
But I'm also curious about the challenges. What are some of the biggest hurdles that scientists face when trying to develop drugs that target Class A GPCRs?
One major challenge is selectivity, as we've already touched upon.
Right.
Remember, there are hundreds of different Class A GPCRs in our bodies, many with very similar structures. Designing drugs that can specifically target one receptor without affecting others is like trying to hit a bullseye on a moving target while blindfolded.
That's hard.
It requires incredible precision and a deep understanding of the subtle differences between receptors.
It sounds incredibly challenging. Are there any other hurdles they have to overcome?
Another challenge is understanding the dynamic nature of these receptors. We know they can adopt different conformations depending on the ligand they're bound to, but we're still unraveling the complexities of these conformational changes and how they influence signaling. It's like trying to choreograph a dance when you can only see the dancers in still frames.
Wow.
We need to understand the full choreography to truly grasp how these receptors work.
This is getting really deep. I'm starting to feel a little overwhelmed by the sheer complexity of it all.
It is complex, but that's what makes it so fascinating.
It's true.
And remember, we're constantly learning more about these receptors and developing new tools and technologies to study them in greater detail. It's a rapidly evolving field with incredible potential for breakthroughs in medicine.
That's incredibly encouraging. But before we delve any further, let's take a short break to let all this information settle in.
Sounds good.
We'll be back soon to wrap up our exploration of Class A GPCRs. Stay tuned. Welcome back to the Deep Dive. We've been on quite a journey exploring the intricate world of Class A GPCRs.
We have.
Before we wrap up, I'm curious about what the future holds for research in this field. What are some of the most exciting areas that scientists are focusing on right now?
Well, one area that's generating a lot of buzz is the development of allosteric modulators.
Allosteric modulators?
These are molecules that bind to sites on the receptor other than the main ligand-binding pocket.
So not the usual spot where drugs typically bind.
I think of it this way. Instead of just flipping a switch on or off, allosteric modulators act more like a dimmer switch, allowing for fine-tuning of the receptor's activity. They can either enhance or inhibit the receptor signaling, depending on the desired effect.
I see. So by targeting these allosteric sites, scientists could potentially have more control over how a receptor responds to a signal.
Precisely, and that level of control opens up exciting possibilities for developing more targeted and precise therapies with fewer side effects.
That sounds promising. Are there any other areas of research that you're particularly excited about?
Another area that's really intriguing is the study of receptor oligomerization.
Oligomerization. That sounds complicated. What does that mean exactly?
It simply means that sometimes instead of acting alone, GPCRs team up and form complexes with other GPCRs.
Okay.
They become molecular partners in crime, so to speak.
So it's not always a solo act.
Yeah.
Sometimes these receptors prefer to work in groups.
Exactly, and we're still trying to unravel the functional consequences of this teamwork.
Okay.
Does it change how they bind to ligands or how they signal?
Right.
Does it affect their overall function?
Got it.
These are some of the questions that researchers are actively exploring.
It seems like the more we learn about these receptors, the more complex they become.
Absolutely, but with complexity comes opportunity.
Right.
The more we understand about how these receptors work both individually and as part of larger complexes, the better equipped we'll be to develop new and innovative therapies.
That's incredibly exciting.
Yeah.
So for our listeners who are now as fascinated by Class A GPCRs as we are, where should they go to learn more? What resources would you recommend?
Well, for a comprehensive overview of GPCRs, I always recommend the GPCR database, or GPCRDB.
Okay.
It's a fantastic online resource with a wealth of information on all known GPCRs.
Wow.
Including their sequences, structures, and ligands.
Sounds like a treasure trove of GPCR knowledge.
Yeah.
And for those who want to delve even deeper, are there any scientific journals that focus specifically on this field?
Absolutely. Journals like Molecular Pharmacology and the Journal of Biological Chemistry regularly publish cutting-edge research on GPCRs.
Okay.
And for a broader perspective on drug discovery, Nature Review's Drug Discovery is an excellent resource.
Fantastic. Yeah. So for our listeners who are ready to take their knowledge to the next level, be sure to check out those resources. This has been an incredible journey into the world of Class A GPCRs. I'm honestly in awe of these molecular machines and the crucial roles they play in our bodies.
I agree. It's been a pleasure exploring this fascinating topic with you and our listeners.
And on that note, we want to thank you for joining us on this episode of The Deep Dive. We hope you've enjoyed this deep dive into the world of Class A GPCRs. Until next time, keep those minds curious.